Okay, welcome back to our lecture in the lecture series over receptors or cell surface receptors, mechanics and functions. Now, as always, I've provided you with a set of learning objectives. They're a good guide to studying for the exam. In fact, the study guides for the lecture exams are just a consolidated place to find all of the learning objectives associated with each lecture. Almost more important than looking at the learning objectives, although those drive what we're talking about in the lecture, is actually having your handout out and available. You can either print it out and take notes by hand and answer the questions and things we go over in the follow along by hand, or you can have it up on a computer or a digital device and you can go through and type in your notes, whatever you're going to do. But I would have that handout out and available to you because that really guides the important points of the lecture that I want you to get. So with no further ado, let's start talking about this. How and when do cells communicate? One of the key elements in all biological systems is cells communicating with one another. So, for example, if a bus is coming at you, you take in sensory information, you process that information, and then you initiate a motor response, contracting skeletal muscles in order to get out of the way of the bus. There are literally, just within that one event, billions of independent communication events that coordinate you as a whole, as an organism, getting out of the way of that bus. So, how do cells talk? How do cells listen? And that's really the basis of what we're going to be talking about today is cell communication. How do cells within the body communicate with one another in order to coordinate this extraordinarily complex set of, biological functions that allow us to maintain homeostasis and live life? So when do cells communicate? Well, one of the places in which there's a lot of cellular communication is during growth. So what you're looking at is embryonic growth in which the lower appendages are forming, and that involves communication among those cells, as does embryonic fetal development, the growth during childhood, right? All of that involves cell communication. Cells need to communicate with one another. They need to say divide, stop dividing, etc. So we communicate during growth. Body maintenance, things like wound healing, or just the general upkeep of the different tissues in your body. So if you look over here, you'll see a guy who scraped his hand, and this is over 30 days. You're looking at that wound heal. Well, what you don't think about when wounds heal is that those involve cells and molecules interacting with one another and communicating with one another in order to coordinate that. So if you look over here at this little GIF over here, you're looking at a wound and you're looking at these cells migrate toward one another and communicate with one another in order to begin the healing process of that wound. Interestingly enough, if you have a wound during the day as opposed to the night, it seems that wounds during the day tend to heal a little bit better because those cells are a little bit more metabolically active. That's just interesting. It's not something you have to know. So during wound healing, during basic maintenance, when, for example, liver cells are dying or the cells in your skin slough off, you need to replace those. We're replacing cells along the lining of our GI tract, our skin, inside our mouth, everywhere, all the time. And that involves communication. There has to be a way to communicate to a cell, hey, you need to start dividing. Hey, you need to start moving toward these other cells in order to seal this wound up, whatever it is. The immune system communicates all the time with other cells. So, for example, you have these cells that go around and they look at other cells and they identify self versus non-self. So they're essentially talking to one another. And the way that they do this is they recognize markers on the surface of healthy cells and they go, oh, that's a healthy cell and it's part of self, and they leave that cell alone, as opposed to, let's say, a cell that's been infected with the coronavirus. This cell may actually initiate a process to destroy that cell, right? So in the immune system, when immune responses are needed, cells are communicating. Now, there's another process called apoptosis, which is programmed cell death. Just like humans have a life cycle in which we grow old, and die, cells also have a life cycle, right? And in order to communicate that a cell needs to die, there needs to be communication. It needs to get a signal to do that. Those signals can be internal or they can be external in the form of things signaling molecules such as hormones. But either way, they're signaled that, hey, it's time to start budding off and getting eaten up by these white blood cells. And there's communication that happens there. So I hate the word. Some textbooks will refer to apoptosis as cell suicide. I don't like that word. It's not cell suicide. It's programmed cell death, right? A cell. There's an appropriate time for a cell to die. All living things will die at some point. So when neurons need to communicate with other different tissues in the body, such as muscles, or with each other, there's communication that happens between one cell and another cell. And this is what we're really going to beat to death when we get into those units over the nervous system and the muscular system, is how does that communication happen? So when you think about the contraction of one of your muscles, let's say like the biceps brachii, for example, that event begins in the brain, and then an electrical signal travels along this neuron. That's a form of communication called an action potential. And it gets to the end of this neuron where it triggers an event at a place called the synapse. This cell is now communicating with this cell. An action potential fires along this cell, and that cell communicates with these muscle fibers, right, which ultimately produces a situation, a response in which the muscle fibers contract. So muscle contraction or skeletal muscle contraction begins in the brain. You have to have cells communicating with one another to get that event to trigger out here, and they do it very quickly. So what you're looking at here is what's called the synapse. You're looking at communication between one neuron and another neuron, and there are all sorts of things involved, and we're going to highlight one of the more important aspects of the communication between cells today. We're just going through what do we use cell communication for. Another system that uses cell communication is the endocrine system. So the endocrine system, this is long-term or long-distance communication. So, and it's also lasts a lot longer, but when you're looking at the secreting cell, this would be like an endocrine cell, right? So a cell in the pancreas, like the beta pancreatic cells, or in your thyroid gland, wherever it is. They secrete a chemical messenger directly into the blood. By definition, any chemical messenger secreted directly into the blood is called a hormone. That hormone then travels through the bloodstream until it reaches its target cell. All target cells have something we're going to beat to death today. They're called protein receptors. Any cell with the protein receptor for that hormone is by definition a target cell. When the hormone binds to that receptor, it triggers a change in the target cell that produces a response. Now, if you remember, our two primary systems of homeostasis are the nervous system and the endocrine system. Now, the nervous system and the endocrine system are capable of detecting changes. So they have receptors. They can detect changes, like in the case of the nervous system, things like is blood pressure getting too high, is temperature getting too high. In the case of the endocrine system, are blood glucose levels getting too high? So they can detect changes, right? They can process that information. So, for example, if temperature gets too high, you have temperature receptors or thermoreceptors in the skin. That information is sent to the brain, and the brain processes that information. In the case of the pancreas or the endocrine system in the pancreas, right, it would be a change in the concentration of blood glucose that's detected by those beta pancreatic cells. So the nervous system and endocrine system can detect changes. They can process that information, and they can initiate responses. Initiating responses is really telling other organ systems and other organs what to do. So what's unique about the nervous and the endocrine system is they have the ability to tell other organ systems and other organs what to do. They do that a little bit differently. In the nervous system, you get really rapid, short-lived responses that are kind of localized in nature. And the nervous system is responsible for that kind of second-to-second regulation. So let's say you need to dilate the bronchi in your lungs because you're running. Well, the information about oxygen status will be sent to the brain. The brain will say we need more air. It'll trigger a situation in which you relax the airways, dilate those bronchi, and bring more air into your lungs. And it'll happen pretty quickly, and it'll be a relatively short-lived response. Same thing with the contraction of skeletal muscle, right? Nervous system detects, processes information, initiates that change. In other words, neurons need to communicate with things like the smooth muscle lining the airways or your skeletal muscles or whatever it is, right? So the nervous system is capable of communicating with other organ systems. So it can detect, process, and respond to changes by communicating with other organ systems. It does this using electrical signals called action potentials and chemical signals, right, at the point of interaction between the neuron and the tissue called neurotransmitters. The endocrine system is capable of doing all that as well. Whereas the nervous system is rapid, localized, immediate responses, the endocrine system is relatively slow, but it affects a wide array of tissues, and the responses last for a much longer period of time. Think those day in and day out things like the regulation of blood sugar, or blood volume, or whatever it is. The point is that the endocrine system can detect changes, process that information, and then release effectors called hormones. And these hormones will travel to different organs and they will tell them what to do, right? So even though the mechanism is different, both the nervous system and the endocrine system are capable of detecting, processing, and responding to changes. One does it very quickly, the other does it relatively slowly, but that's why we call these the systems of homeostasis. Now, when you think about what we're going to be focusing on today, we're going to be focusing on how one cell communicates with another cell in a very basic way. So here we have a point of communication between one neuron and another neuron. And the point of communication between one neuron and another neuron is called a synapse. At the synapse, there are chemical messengers released, right, from the cell doing the communication. So you can kind of think of this as the cell talking. And those particular chemical messengers bind to protein receptors in the membrane of the cell that's receiving that information. You can think of this as the cell that's listening. So the cell releasing the chemical messenger, right, or the ligand is the cell doing the talking. The cell receiving that is the cell doing the listening. And then that will trigger some kind of response. When you hear the term ligand, it means chemical signaling molecule. It's a blanket term for chemical signaling molecule. The chemical signaling molecules, or ligands, used by the nervous system are neurotransmitters. We refer to the ligands, or chemical signaling molecules, used by the nervous system as being neurotransmitters. So neurons communicate with other cells or with each other using neurotransmitters. The endocrine system, on the other hand, there's a change. Hormones are released into the blood. It's not localized communication. It's not short distance. It's long distance communication. These hormones travel through the bloodstream where they eventually exit the bloodstream, bind to receptors on cells all over the body, right? Any cell with a receptor for a hormone is a target of that hormone, and they trigger this kind of widespread change. It's slow. It takes a lot longer to happen than what happens with the nervous system. But they trigger this kind of widespread change and prolonged response. And the chemical signaling molecules that are used by the endocrine system are called hormones. In fact, any chemical signaling molecule secreted directly into the blood that then travels to a distant target organ. So this would be an example of hormones released by the pituitary gland traveling all the way, for example, to the ovary or the testes or to the adrenal gland, right? Any chemical messaging molecule released directly into the blood is called a hormone. The two ligands we're going to focus on most in this class and in AMP2 are neurotransmitters, which are how the chemical messengers used by the nervous system to communicate with other cells. So it's neurotransmitters are what neurons use to communicate with other cells, and hormones are what endocrine glands use to communicate with other cells. What makes the nervous system and endocrine system unique, and why we refer to those systems as being systems of homeostasis, is that they're capable of detecting, processing, and responding to changes by telling other organ systems what to do, right? They can tell other organs or other cells what to do, so they're adept at communicating, because that's ultimately what you're doing. Those are kind of processing centers that communicate the information about change to other tissues, right, in your body, or other cells that make up tissues, that make up organs, and it's that triggers the response or the change. So when you hear the term ligand, it's a blanket term for chemical signaling molecules. Neurotransmitters are the chemical signaling molecules of the nervous system. Short-lived responses, rapid, right, locally activated, meaning that it's a short distance communication when a neuron communicates with another cell. Endocrine system takes a little bit longer, but it's a widespread response because these hormones are getting directly into the blood. It's long distance. All of these cells are targets, can trigger massive change. hormones are the chemicals that the endocrine system uses to mediate its responses. Now, in order for either the nervous system or the endocrine system to affect a cell, that cell needs to have a protein receptor. And we've had the lecture over gene expression. So we know DNA is information for making RNA. That process is called transcription. RNA is information for making proteins. That process is called translation. Proteins go out and serve some kind of function. They're machines. A very important class of proteins are what are called protein receptors, right? Now, when you think about an extracellular receptor, notice that, so this would be the plasma membrane. Here would be your phospholipid bilayer, and here's the protein embedded within the membrane. You have your signaling molecule. This could be either a hormone or a neurotransmitter. Signaling molecule binds to the receptor. You have step one in what protein receptors do. They receive information. Now, they transmit that information from the outside to the inside of the cell. How does a hormone floating around in your blood ultimately produce changes within the cell? Well, there needs to be a receptor to bind to that hormone and trigger some kind of internal response. So there's reception. Then you have transduction. This is where a series of what we call relay or secondary signaling molecules become activated. And along each step of activation, there's usually amplification, meaning more and more of these molecules become activated. I'll use an example of what happens during the transduction pathway in many pathways. Let's say that you told me, I'm the receptor, I need to clean the house. And then I told my 3 nephews, and they told their 3 cousins, and then they told their 3. At each step, you get more and more people cleaning the house, and ultimately, you can produce a massive response from very little signaling molecule, and it will produce some kind of change in the cell, whether it's burning glucose faster, or making glycogen, or storing lipids, or synthesizing proteins, whatever it is. So those are always the 3 steps. Receptors function as an intermediate. They allow communication to happen. They're kind of like the protein ears. So they mediate communication between the outside and the inside of the cell, or even internally, and we'll talk about that in just a little bit. But there's always 3 steps to this pathway. So how does insulin lower blood glucose levels? Well, it binds to an insulin receptor, it triggers transduction, and there's a response, and that is the cell sucks up more glucose. Right? They're all, you could just go on example after example, but this is the basics of what receptors do. They receive information, they activate molecules in the cell. That process is called transduction. Eventually, a protein will be activated that does something, right? And that's called a response. And in that doing something could be, synthesizing glycogen or synthesizing lipids or breaking down, the fatty acids, whatever it is. Now, when you think about extracellular receptors, when I say the term extracellular receptor, I mean a receptor embedded in the plasma membrane, not in a cell membrane, but in the plasma membrane, that barrier between the internal and external environment. Extracellular receptors always have a portion that's facing the extracellular fluid, or ECF, and they always have a portion, right, that's facing the intracellular fluid, or ICF, and that's how they can bring signals from the outside of the cell to the inside of the cell as they span or they traverse the entirety of the phospholipid bilayer. Now, 2 types of extracellular receptors we're going to focus on for just a minute are ligand-gated ion channels. Remember, a ligand is a blanket term for a chemical signaling molecule, so this could be a hormone or a neurotransmitter. We haven't specified it. Now, what ligand-gated ion channels do is when a ligand binds to its receptor protein, this is a class of receptor, and both of these 2 classes we're going to look at initially are considered extracellular receptors because they're embedded in the plasma membrane and they have portions contacting the ECF and portions contacting the ICF. But what ligand-gated ion channels do is when a ligand binds to this receptor, it triggers a conformational or a shape change in this channel that transitions it from a closed to an open state and allows particles such as ions to flow through. So we'd call this a ligand-gated calcium channel. The ligand-gated calcium channel is really a type of receptor. You find them in places like dendrites and neurons that are responsible for receiving information. If a ligand-gated calcium channel allows the movement of calcium, it's specific for that. It doesn't allow the movement of any other ions. So these ligand-gated ion channels display specificity not only for the ligand, but for the ions that they allow. But really, they're just ligand binds, either neurotransmitter or hormone. We'll focus on neurotransmitters. Channel opens, ions are allowed into or out of the cell, which triggers some kind of response. G-protein coupled receptors, on the other hand, are different. They always have 1, 2, 3, 4, 5, 6, 7 transmembrane spanning regions. And G-protein coupled receptors, there are a lot of things. Hormones, neurotransmitters that bind to these. The binding to this receptor activates a G-protein internally in the cell and triggers some kind of cell pathway. So it's not a direct ion channel. So whereas these guys, these receptors, are really fast acting, they're short-lived, but they produce immediate responses. These guys are a little bit slower, but they can produce much larger responses. So two types of receptors you should be familiar with that I'm going to refer to as extracellular because they're embedded in the plasma membrane are ligand-gated ion channels and G-protein coupled receptors. Now, when you think about ligand-gated ion channels, let's think about one neuron communicating with another neuron. So here's the neuron doing the communication, and here's the neuron receiving the communication. Right now, as you're watching this, billions of neurons in your brain are communicating simultaneously. So they communicate at points called synapses. And what essentially happens is an electrical signal gets to this point called the synaptic end bulb. You don't have to know this quite yet. Triggers the exocytosis of these little synaptic vesicles that contain neurotransmitters. So this is the cell doing the communication. Neurotransmitters are released. Many neurotransmitter receptors are really just ligand or ligand-gated ion channels. So when the neurotransmitter, if you see here, binds to that ligand-gated ion channel, it opens up, right, a channel. So a channel goes from being closed to open, and if that's a sodium channel, for example, sodium's going to rush into this little postsynaptic cell, and it's going to trigger that cell to depolarize, which tells that cell, hey, you need to communicate with another cell really quickly, something we'll get into. But ligand-gated ion channels are really important in the way that neurons communicate with one another. And they're a type of receptor, right? So when I say a ligand-gated, the GABA receptor is really a ligand-gated chloride channel, you kind of have an understanding of what I mean when we get further into that. Another type of extracellular protein receptor we're going to get into are G-protein coupled receptors. You find them in both the nervous system and the endocrine system, but we're going to focus on the endocrine system just for the sake of this. So let's say that this cell here is an endocrine cell like a chromaffin cell that releases epinephrine, right? Epinephrine is released into the blood. Any chemical messenger released directly into the blood that's transported to a distant target cell is by definition a hormone. That epinephrine travels through the blood, it circulates through the bloodstream, and then it binds to receptors on distant target cells. Any cell with a receptor is a target. So epinephrine has a wide array of targets, right? It can work on a lot of different cells in the body. So it binds to distant target cells where it triggers some kind of response. Now, that's not the only hormone we can talk about, but it's, we could talk about insulin, we could talk about glucagon, we could talk about a range of well, thyroid hormone wouldn't actually be one of these, growth hormone, things like that. We could talk about a whole range of hormones, right? So G-protein coupled receptors. G-protein coupled receptors are proteins embedded within the membrane of cells that activate secondary signaling pathways, and in each step of the secondary signaling pathway, you get a process known as amplification. So let's look at what that means. Let's say we have a water-soluble hormone. Water-soluble hormones are polar molecules, so they can't get directly across the phospholipid bilayer, so they need to bind to extracellular receptors in order to transmit that information from the outside to the inside of the cell for that initial reception step. Lots of different water-soluble hormones. We'll think of protein hormones as being water-soluble hormones, or peptide hormones as being water-soluble hormones, right? So you have these peptide, aka protein hormones, and they bind to this G-protein-coupled receptor. This receptor is linked to another protein called the G protein. That's why it's called a G-protein-coupled receptor. When a hormone binds to this G-protein-coupled receptor, it activates a G protein. That G protein then goes and activates a molecule in the pathway, one of your transduction molecules or secondary signaling molecules called adenylyl cyclase. Adenylyl cyclase converts ATP into another molecule called cyclic adenosine monophosphate. For every hormone that binds, you get the activation of one G protein, one adenylyl cyclase. Each adenylyl cyclase can convert, let's say, 1,000 ATPs into cyclic adenosine monophosphates. So from a single molecule binding, you now have 1,000 activated secondary signaling molecules in the cell. That's called amplification. Cyclic adenosine monophosphate gives up a phosphate to a protein kinase, activating it. In other words, it turns protein kinases on. Now I want you to tune in here. Let's say you had 1,000 activated camps, and every one of those can activate 1,000, protein kinases or whatever it is. You've now gotten amplification along the pathway. So you now have even more activated molecules within the cell. Protein kinases are really important because what protein kinases do is they turn functional proteins on. They do this through a process called phosphorylation. Phosphorylation is taking a phosphate and pinning it to another molecule. So stripping that phosphate from ATP and pinning it to a protein turns a protein on. From that single ligand binding, that single hormone up here binding, you get millions of activated proteins in the cell that can produce a whopping physiological response, whatever that physiological response is. Maybe it's breaking down glycogen, maybe it's building up lipid reserves, whatever it is. You can get a whopping physiological response. So protein kinases turn other proteins on. So let's say I was a protein kinase and there are 10 people in my living room who are responsible for vacuuming because I got a gigantic living room, right? There are some times when they're not supposed to be vacuuming, and then I go around and I turn on their vacuum cleaners, and that's what activates their vacuuming activity. I'm like the protein kinase, and I'm turning on a functional protein that's actually going to go and do something. It's like a manager telling another protein what to go and do, and it turns it on, and then those proteins go out and physically exert that physiological response. So let's look at an example from an incredibly talented educator. I love this guy who does Bozeman science, but let's look at an example pathway. So here we have a hormone called epinephrine, and one of the physiological effects that epinephrine can have is it can elevate or increase blood glucose levels. So let's say you're in fight or flight mode, you release epinephrine, aka adrenaline, into the blood from your adrenal glands, and because your body wants more glucose so it can burn immediate energy reserves. That epinephrine is going to bind to this G protein coupled receptor, which is going to activate the G protein, which is going to activate adenylyl cyclase, which is going to convert ATP into CAMP. That CAMP is going to activate your protein kinase, which is going to activate the protein that's responsible for breaking glycogen down. Remember, the building blocks of glycogen are glucose, so when you break glycogen down, it releases glucose into the blood. There's a mechanism here. So let's say that you're running away from a scary thing coming at you, right? And you need to increase your blood glucose levels. There's a lot of different communication events, but ultimately your adrenal glands, and specifically your adrenal medulla, these cells called chromaffin cells, release epinephrine into the bloodstream. That epinephrine travels around until it gets to target cells, and the goal, what you're wanting, is to increase the amount of glucose in the body, free sugar that we can use for energy.
Into the cell, but it's going to attach to the receptor. So this is called the receptor. Do you remember what that's called? That's right. It's called the G protein. And so what will happen is that ligand will attach to the G protein. It's got a number of different subunits. This one right here on the end is called the alpha subunit. That's
Don't worry about knowing.
Hey, if you're not getting any of these, you may want to go back and watch the earlier portions of the video. We've got this over here, and this is probably the one that you're going to struggle with the most. What's the name of that? Oh, that's right, adenylyl cyclase. And so what happens is the alpha subunit is going to attach to adenylyl cyclase. So we then have an enzyme that's functioning.
Receptor
it's going to take in these little starbursts. Those aren't starbursts, they're called. That's ATP. Right, ATP. ATP will then be converted to cyclic AMP or CAMP. And cyclic AMP are now going to go work on this protein kinase. That's right, protein kinase. It has two portions that are catalytic and two that are...
Don't worry about catalytic.
Regulatory. That's right. Okay, so the cyclic AMP is going to move over to that. But let's watch what happens for a second. Because there's not just a few cyclic AMPs in a cell. There are going to be lots of cyclic AMPs and lots of protein kinases.
Amplification.
And so remember when we talked about our analogy of Jimi Hendrix, this is where we can amplify the message. So we just have this one ligand up here, but we can have all this action going on. I didn't want to animate all of that, so I went back to just one protein kinase. So we will free up the catalytic portions.
Don't worry about that.
We now add energy to them.
Kinase.
What's that called? Phosphorylation. That's right. We're going to add a phosphate group to them. They're going to turn yellow, and now they're able to pass on that phosphate group to phosphorylase. So it's activated, and it can break down glycogen.
Glycogen's broken down, individual glucose molecules.
So that's the signal transduction pathway. It's fairly simple. It's got a lot of steps in it, but it's just like playing the electric guitar, and so I hope that's helpful.
Those glucose molecules can be used for immediate energy, right? So that's a G protein coupled receptor. Those are the two extracellular receptors you need to be familiar with. We also have what are called intracellular receptors. Intracellular receptors are receptors located inside the cell. So lipid-soluble, aka non-polar signaling molecules such as steroid hormones like testosterone, estrogen, progesterone, aldosterone can get right across that plasma membrane. And because they can get right across that plasma membrane, we actually, in order for them to trigger some kind of pathway or response, you need the receptor internal or inside the cell. So you think about testosterone. I'll focus on testosterone. You do some resistance training, and you need to build up more muscle. You need to recover from that. So the resistance training, right, sends a signal to the brain, which triggers the anterior pituitary gland to release a hormone that tells the testicles to release this guy right here, the most important of male hormones, testosterone. Testosterone then binds to receptors on the surface of, binds to receptors within the cell in things like skeletal muscle fibers, triggering protein synthesis, leading to bigger muscles over time. Now, intracellular receptors don't work nearly as quickly as extracellular receptors. Extracellular receptors are activating pre-existing proteins. Intracellular receptors, such as steroid hormones, the receptors that bind to steroid hormones, are a little bit different. So let's pretend this is the testosterone example. Now remember that blood, and specifically blood plasma, is predominantly water. It's what's called an aqueous solution. It's like 93% water. So in order to dissolve in blood plasma, you have to be polar. A molecule has to be polar or carry a charge. Steroids are nonpolar. They're classified as lipids, so they can't dissolve directly in the blood plasma. So when they're traveling around the body, they actually have to travel bound to what are called transport proteins. The transport protein that moves testosterone around is called sex hormone binding globulin. You don't have to know that, but that's what it's called. So whenever you hear bound versus free, for example, bound versus free testosterone or bound versus free estrogen, when you're talking about bound, you're talking about it being linked to a transport molecule. And when you're talking about it free, that's the steroid that's available for use by the body. That becomes really important when you start doing diagnostics on things like low testosterone levels. So let's say it's testosterone. Steroid hormones released at the target cell, because it's lipid soluble, it crosses right across that phospholipid bilayer and it binds to a receptor inside the nucleus. This is called an intracellular receptor. That receptor is really, right, a transcription factor. In other words, it turns a gene on. When that gene is turned on through the process of transcription, you'll make mRNA. Through the process of translation, a new protein will be made. That new protein will alter the cell's activity. So let's go back to why extracellular receptors work more quickly than intracellular receptors. Extracellular receptors are activating pre-existing proteins. It's like turning things on, right? Intracellular receptors are changing gene expression, and that gene expression is making proteins from scratch. So do you think it would take more time to turn 10 vacuum cleaners on or to make 10 vacuum cleaners from scratch if you were given the parts? Obviously, to turn them on. That's what extracellular receptors are doing. Intracellular receptors are creating proteins from scratch, not activating pre-existing proteins. So those intracellular receptors trigger the expression of a gene, which ultimately makes a protein. Let's go back to our testosterone example. So let's say you're doing weight-bearing exercise and you get done doing weight-bearing exercise. There's communication between your muscles and the brain. The brain tells the pituitary gland to release luteinizing hormone, which tells the testes to release testosterone. That testosterone then travels to, let's say, a skeletal muscle cell, where it goes into the cell and it binds to an intracellular receptor. That intracellular receptor activates a gene that makes one of your key contractile muscle proteins called myosin. DNA to RNA, RNA to protein. That protein's job is to generate force during muscle contraction. More of that protein means more force during muscle contraction. Your muscle grows or hypertrophies. And that's the mechanism. There's a mechanism to all of this. So those are intracellular receptors. Only lipid-soluble hormones like steroid hormones are going to activate intracellular receptors. Now, when you think about pharmaceutical application, you're going to hear, when you take pharmacology, you're going to hear a lot about receptors. You're going to get inundated with receptors because pharmacology is just applied physiology. And when you're hearing receptors, now you know, okay, these extracellular receptors are embedded within the plasma membrane of cells throughout the body, right? And intracellular receptors are inside the cell. And receptors essentially turn signaling molecules into responses inside the cell. They trigger some kind of event inside the cell. So when you're thinking about a natural chemical, let's call this a neurotransmitter, right? You have that natural chemical, it binds to its receptor, it triggers the intracellular activity. This would be an extracellular receptor because it's embedded in the membrane. Agonists, aka stimulatory medicines, activate those receptors and they enhance the activity of the natural chemical, whereas antagonists inhibit those receptors, preventing the activity of the naturally produced chemical, blocking the cellular activity. So when you hear the term stimulatory medicine or agonist medication, you know it activates a receptor, and antagonists inhibit a receptor. And in doing that, they change the intracellular activity in the hopes of having some kind of therapeutic effect, making you feel better. So when you think about receptors for medications, cells that are really sensitive, let's say you're really sensitive to certain classes of medication, it probably means those target cells that medication is activating have lots of receptors. As opposed to, let's say you're not active, let's say I'm very sensitive to caffeine and somebody isn't, right? Somebody else is not. And we say, oh, I can drink like 5 cups of coffee and you can't. Well, it probably has to do with receptors, right? I probably have a lot of them and I'm very sensitive to it, where you don't have very many of them and you're a little bit less sensitive to it, or you've downregulated that sensitivity over time. If there are no receptors or different receptors, you're going to get no response to that medication. Many of your medications, not by all of them by any stretch of the imagination, but many work on receptors. So there's an idea cells are able to regulate what's going on. So prolonged exposure to high concentration of an agonist. Let's say you take a medication or you use a drug that increases dopamine in the brain. Initially, you had a really, like, let's say Wellbutrin or something like that. Initially, you have a really strong response. But over a prolonged period of time using that particular medication, the cell goes, ooh, we're getting dopamine all the time, and it reduces the number of receptors available in the cell. Well, that's the what accounts for things like tolerance to medications, or sometimes medications just poop out like antidepressants. It's because the cell is regulating its activity, it's calibrating its activity to what's going on. Upregulation, on the other hand, if you introduce an antagonist, and that antagonist is always there, the cell's going to produce more receptors, so you're probably going to have to take more of the medication because it's trying to calibrate its activity. So think about that up and down regulation, and once you kind of understand that agonists activate receptors and activate those pathways associated with those receptors, and antagonists inhibit receptors, you can learn a lot about medications because they're essentially classified as agonists and antagonists of receptors, huge classes of them. So when you hear like GABA agonist, well, a GABA receptor is really just a ligand-gated chloride channel. When anything that activates a GABA receptor binds to that receptor, it opens up these chloride channels. Chloride, which carries a negative charge, rushes into the cell. The cell becomes more negative, and it essentially turns the cell off. GABA is an inhibitory neurotransmitter. We haven't gotten there yet. You don't have to worry about that yet. So whenever you hear GABA agonist, you go, oh, that activates GABA receptors. GABA receptors inhibit neurons. They're downers. Benzodiazepines, barbiturates, ethanol, which is the type of alcohol you drink, topiramate, which is a migraine medication, the seizure medications, and all you have to go is it's a GABA agonist. Oh, it's a downer. It's a downer. It's a GABA agonist. I don't even need to know the rest about it. In general, it's a downer. I get it. Right? And if you study it, yeah, you're going to have to elaborate on that, but it's important to kind of understand that. So with that said, we're done.